For us to appreciate antimicrobial resistance and to fight the emergence and spread of resistance, it is important for us to understand the mechanisms of antimicrobial resistance. And this can be ideally attained through two mechanisms. One is innate or intrinsic, and these are specified by naturally occurring genes that are found in the host chromosome. The second mechanism is acquired, and this involves the transfer of resistant determinants or genes that are born on plasmids or bacteriophages and other mobile genetic material. And the transfer of this genetic material occurs through conjugation, transformation, and transduction pathways. And we are going to look at each of these. In intrinsic or innate mechanism for antimicrobial resistance, the microorganism can either destroy or inactivate the antimicrobial agents. The microorganisms can also have efflux systems that will effectively pump out any antimicrobial agent that gets into their systems, or the microbes can undergo DNA mutation and ideally evade any targets that the drug could be utilizing to kill the microorganisms. In acquired antimicrobial resistance, there is gene transfer from one cell or from one organism to the other. And this can either happen through conjugation, transformation, or transduction. In destruction or inactivation by the microorganisms, many bacteria have the ability to produce enzymes that will chemically degrade or deactivate the antimicrobial agent, and this effectively renders them ineffective against the bacterium. The antimicrobial agent is either degraded or modified by enzymatic activity before it can reach the target site and damage the bacterial cell. A good example is penicillinase, which is produced by staphylococci that degrades penicillin. The other mechanism which is effective for the microorganism is the efflux system, where the bacteria, through a channel termed as a porin, can pump back out the antimicrobial agent by the efflux pump. Bacteria increases efflux to prevent intracellular accumulation of the antimicrobial agent necessary to exert their lethal activity inside their cell. Membrane permeability can also be altered through regulation of porins, which are the gates into a cell. The next mechanism of resistance is mutational resistance. And mutation is literally a change in the DNA. And this can sometimes result in a change in the gene product, which is the target site for the antimicrobial agent. So if the target site changes, then the antimicrobial agent is not able to exert its effect. It's more like changing the lock and using the same key to open a door. The next level of resistance or acquisition of resistance is acquired resistance. And this can happen in different ways. And normally would occur by gene transfer. And this involves the transfer of resistance through mobile genetic material. And this can occur via conjugation, transformation, or transduction. The DNA material is shared by a plasmid, which is moved from one cell into another. In conjugation, two cells are normally in close proximity to each other, and there is a hollow bridge-like structure which is known as a pilus that forms between the two cells. Bacteria would form a bridge through which a copy of the plasmid bearing the resistant gene is passed from the donor to a recipient. Plasmids generally carry the genes that confer resistance to antimicrobial agents, and therefore any susceptible bacterium can acquire resistance. And this may be the mechanism for enterococcal resistance to vancomycin. In transformation, the transfer of a naked DNA from a dead bacteria to other bacteria in close proximity occurs, and the living bacteria is able to incorporate the resistant gene into their own DNA. Some of the free DNA may contain advantageous genes such as antimicrobial resistant genes and may benefit the recipient cell, essentially making it resistant to similar antibiotics that it might be exposed to. For transduction, bacterial DNA is transferred from one bacterium to another inside a virus that potentially infects bacteria. And these viruses are referred to as bacteriophages or phage. And the DNA that's transferred from one bacterium into another may contain resistant genes.